Hi, welcome. I hope you're doing well. What we're going to be doing is looking at data on COVID RNA sequences. So this is going to be a really fun one. I was pretty excited to see that this data was actually available. So I'm going to start off with a little presentation just because there are a few complexities to what we're going to be working through today. So our goal today is going to be to download and analyze RNA sequences of COVID and two of its major variants, Delta and Omicron. So the first thing we need is the metadata. So that is all of the COVID RNA sequences that are out there, and basically an ID that lets you download the actual sequence. And then we need to actually download the sequences. So those are the two steps we're going to follow today. Then we're actually going to do a little bit of analysis on the sequences. And before I do that, just a quick overview of RNA and what it is. So RNA is the source code for COVID. So the COVID virus looks something like this, and RNA is basically inside an envelope. And what RNA does is it enables the virus to enter a cell and then replicate itself. So a COVID virus will enter your cells, go to the cell nucleus, and then replicate itself, and then continue replicating afterwards. And the RNA is the code that basically enables one COVID virus to build another COVID virus and thousands more. So RNA, basically, you can read the RNA and you can build proteins and then eventually build the COVID virus. And if you're familiar with DNA, DNA is human source code, and DNA consists of nucleotides. So a DNA sequence will consist of these four nucleotides: thymine, cytosine, adenine, and guanine. So if you ever read a human DNA sequence, it looks like T, T, A, G, C, etc. DNA has two strands. RNA is very similar to DNA, except for two key differences. It only has one strand. So instead of the two strands of DNA, RNA only has a single strand. And thymine is swapped for uracil. So these are just different nucleotides that are kind of the base of a DNA or RNA sequence. So what we're going to do today is we are going to try to get an RNA sequence, which will look something like this. GC, it won't be U, it'll actually be T, because it just how it's displayed and stored, the convention is to use a T instead of a U. So it will basically get this long RNA sequence for COVID and its major variants, and then we can compare them. And we'll be working with data from the NIH. The NIH is a federal government agency in the US that sequences data and stores it, sequences RNA and DNA data from around the globe and stores it in a searchable database. We are going to be using data sets available from the NIH. And if you do want to kind of just dig deeper and explore more data that the NIH has on COVID, you can check that out here. So the actual data set we're going to be working with today is this NCBI datasets.csv file. And there's a few columns that are important that I've included here, but there are other columns that we'll see in the data when we load it. Each row is basically metadata for an RNA sequence. So for example, this first row is the first RNA sequence that was ever sequenced from the COVID virus. And it's called the reference sequence, or the RefSeq in the sequence type. It was sequenced in China. The RNA was collected in December 2019. And the name of the sequence is here, and the ID that we can use to access it is here. So this is the data set we're going to be working with. We'll basically pick some sequences that we want to dig into more, and then we'll download the actual sequences, which will be this, GCT, ACG, etc. All right, let's go ahead and dive into it. So I'm going to be using Jupyter Lab for this, and we are going to also be using pandas and a library called biopython, which I'll show you how to download later. So the first thing we're going to do is we're going to import pandas as PD, and we're going to read in this metadata, which is this NCBI datasets.csv file. So that's the file. Let's take a look at the first few lines of the file just to make sure it looks like we expect. So I showed you just a short table with only a few columns earlier. But you can see here it's very similar, just there's some extra columns. So this first row is still the reference sequence. Another interesting one is nucleotide length. So this is the number of nucleotides in the sequence. So we can see the reference sequence has 29,000 nucleotides. So each nucleotide is what you saw, ACTG, one of those. All right, so now that we've read in our metadata, we can go ahead and get started analyzing it. So we'll just do some quick exploratory data analysis on the metadata before we actually download the sequences. So we'll take a look at the shape. So we can see we have 847,000 different sequences, or metadata for different sequences. We don't actually have the sequences. So we want to narrow that down so we can make sure that we only analyze a few sequences. So to help us do that, the first thing we'll do is we'll turn the collection date into an actual date field. This just makes it easier to work with. Right now it's actually a string. And this is just the date that the sequences were collected. So we can use the pandas toDateTime function to do that. And what we'll do is we'll just say metadata collection date. So we'll pass the collection date into that function, and it'll convert it from a string to a datetime. So that's done now. And we can verify by checking metadata.dtypes. And you can see that we now have a datetime for collection date, which is exactly what we wanted. The next thing, this is my personal preference, it may not be yours. I really like to avoid uppercase in column names and spaces because I just don't like typing them out for whatever reason. You may have different preferences, but I'm just gonna convert the column names to make them easier to type out. So we're gonna replace the space with an underscore. And basically, metadata.columns gives you all of the column names in the metadata data frame. And what we're going to do is a list comprehension that iterates over every single column name. And it's basically going to lowercase it and then replace the space with an underscore. So let's go ahead and do that. And now if we take a look at metadata, we can see our improved column names. Again, this is a personal preference. You may be totally fine typing out the column names that existed, so you may not need to do this. So next, let's look at the continent column. So let's take a look at metadata. Sorry, let's create a continent column. So if you can see, geolocation is a combination of multiple locations at the moment. So continent and country, and in some cases, city as well. So what we want to do is just get the first piece, which is the continent. So we're going to remove everything that comes after the semicolon. Geolocation. And then we'll use the pandas string accessor and the replace method, which basically runs a replacement on every single item in a column. So this is a regular expression. This says replace the semicolon and anything after it with nothing. So essentially delete it. And we'll tell pandas to use a regular expression. All right, so what this will do is this will go through every item in the geolocation column. It'll find a semicolon, and it'll remove the semicolon and anything after it. So we'll be left with just the continent. So let's go ahead and run that. And take a look at metadata. And we can see continent is now just the continent. We removed all the other stuff from geolocation. All right, now what we can do is we can look to see the first COVID sequence that was sequenced on each continent. So we can group the data frame by continent, and then we can apply a function to basically only take the first row. So what we'll do is we'll first sort the values by the time, so the collection date, and we'll just take the first one in each group. All right, so this tells us in each continent when the first sequence that was collected in that continent. So in Africa, the first sequence was collected 2020, February 6th. In Asia, it happened earlier in 2019. And in Europe, North America, South America, it was 2020. The dates are a little bit fuzzy here because the original collection date column, in some cases, only had a month. We turned it into a date, and in that process, it added a day, and it defaulted to the first day of the month if there was no day. But things that were collected January 31st will now appear as having been collected January 1st. So there's a little bit of uncertainty there if this actually was the first, but close enough for our purposes. This will show you the first sequence, or close to the first sequence, that was collected in each continent. Okay, so the next thing we can do is we can see how many sequences were collected by continent. So what we can do is use the pandas value counts method to be able to see exactly how many sequences were collected in each continent. So in this case, this was an NIH dataset, which is an American institution. So as you expect, North America and Europe are very heavily represented. But there are other institutions around the world that collect COVID DNA sequences, and it may look very different in those institutions, the distribution of how many sequences per continent they've collected. All right, next let's look at nucleotide length. That's another column, and we can actually run the pandas describe method to figure out what the longest nucleotide length is and what the shortest is. And nucleotide length is what I showed you earlier. It's how many nucleotides are in the sequence. So each of these is a different sequence that was collected and then sequenced, so you can see the actual source code of the virus. And you can actually see what the variation was in the virus over all the samples. So you can see it's not very high. This one is a little bit weird, but in general, the COVID virus is around 29,000 nucleotides long, the COVID RNA sequence. Let's take a look at this. This is kind of a weird outlier. And then we will just say, we'll find basically the row that is the minimum. So what this is doing is it's saying search metadata for any row where the nucleotide length equals the minimum nucleotide length, which in this case is 2859, which is weirdly low. All right, so if we look at this row, we see that the completeness column says it is a complete genome. So this is probably just a typo, and they meant to put an extra 0 or some other digit in here. All right, the last piece of exploratory data analysis we can do is we can look at how many samples were collected by month. So first we'll create a sample month series. And what we'll do here is we'll say metadata collection date. And we'll basically just truncate it. So what we'll say is .values.asType, and this basically changes the type of the column. But we're going to pass in a special code. Don't worry too much about this code. This isn't a thing that comes up very often. But what this is doing is it's converting it to a specific kind of date value. And this date value is basically just year and month. So it's cutting off the day. And the reason we're doing this is it's going to make our sample, or plotting how many samples are collected by month, just look a little bit nicer. So now what we can do is we can call value counts to just summarize how many samples were collected in each month. And then we'll sort it, and then we'll plot it. All right, so what this shows us is on the x-axis, each month from when the COVID outbreak first started through to now. And it shows us how many samples were sequenced, how many RNA samples were sequenced and put into this database by month. So we can see activity increased in 2021. It increased again recently. Unclear why. Maybe if you work with genomics, you know why, and there's a good reason here. But it is interesting to see those trends. All right, so that's the exploratory data analysis piece. You could do a lot more exploratory data analysis if you wanted to. There's a lot of interesting things you could find here. But for now, I'm going to move on to actually analyzing the sequences themselves, not the metadata. All right, so to do that, we first need to find some sequences that we want to analyze. So the first thing we're going to do is we're going to pull out the reference sequence, which is the first COVID genome that was fully sequenced. And it's called the reference sequence. So the sequence type column is going to be reference sequence. And that's this one. So what we need here is actually the ID, so this nucleotide accession. So we're going to stick this in a list, just nucleotide IDs, I guess, is what I'll call it. So that's going to be the first one. And that's just the reference sequence. So now let's find the metadata for some other common variants. So we're going to look specifically at Delta and Omicron. So we'll look now at Delta, and what we'll do is there's this isolate name column, and this contains information about which variant it is. So in the case of delta, there's actually a little label that says delta. So what we'll do is we'll search this column for the string delta. And the way we'll do that is we'll say, okay, isolate name. We'll use the pandas string accessor to use the contains method. So basically this will search through every string in the row and say, does this string contain delta? And if it does, it'll return true. If it doesn't, it'll return false. Now, some of the isolate names are missing. And in that case, we want to fill NA with false. So we'll just run that just to make sure that we don't get tripped up by missing values. All right, so these are the sequences of the delta variant. There's probably more sequences of the delta variant, but these are the ones that are specifically tagged delta. So you can see in the isolate name, they all have a little delta tag. So we'll look at one of these to help us look at the actual delta sequence. Next, we'll look at Omicron. And to do this, I'll just copy and paste and switch this to Omicron. So let's run that. All right, and we can see we have one Omicron sequence as well. All right, so for delta, we're gonna grab, let's see which one we wanna grab. We'll grab this sequence because this is tag delta 1. So let's go ahead and grab that. So that'll be our delta sequence. And then we'll grab this as our Omicron sequence. Okay, and then what we'll do is we'll go back and grab the first sequence from North America, just so we have another base sequence to look at, just to see how that compares to the reference sequence. So these are going to be the 4 sequences that we download and work with. And I'll rename this to sequences. All right, and then just the human names for this, I'll just create a list, are going to be reference. So the first is the reference sequence, then we have the base sequence, then we have delta, then we have Omicron. So those are the 4 sequences we're going to work with. And what we can do is we can then just look at the metadata for those sequences. And the ID is called this very long name of nucleotide accession. And we'll basically look for any rows in metadata where the nucleotide accession is in this list. Okay, and then we can just take a look at our selected sequences. All right, so these are the sequences we pulled out. Okay, the next thing we're gonna need to do is go ahead and download these sequences. So let me jump back to the presentation real fast. And let me show you how we do that. Okay, so basically what we need to do now is we have the nucleotide accession numbers. We need to actually get the sequences, which look like this and are about 29,000 characters long, given the length of the nucleotide sequences. All right, so the way we're gonna do this is we're gonna use a library called biopython, which has a really handy method which helps you look up RNA sequences and DNA sequences by accession number and then download them. So I'm gonna show you how to install biopython. It is pip install biopython. Not a lot of drama there with the name and how to install it. And the import is a little bit different than other libraries. So you're actually going to import bio, not biopython. So just bio. And what we're going to do is import I think it's pronounced Untrez, but this is the actual module that does the search for us. And we need to set an email. So I'm going to set my email here. Because whenever you download a sequence, it sends your email to the NIH. It's just a nice thing to do to let them know who you are and let them contact you if anything is weird with how you're accessing the site. So we're going to create a function called download sequence. And basically what this will do is, given this nucleotide accession, it will download the actual sequence and give it to us. So the first thing we'll do is we'll create a handle. So this is a search handle. So this Untrez module in biopython has this eSearch function, which will search the NIH's database for a given sequence. So we're going to tell it to search the nucleotide database, and we're going to tell it to search using our ID code, which is just this nucleotide accession number. And we're going to say only return a single value. So ret max is 1. And then we're going to read from that search handle. We're going to read the results. And then we're gonna do another search. You have to do 2 searches with this module. So we'll again search the nucleotide database, and then we'll pass in our ID, which is just, this is just internals to the library, but basically this record, when we ran our first search, will have another ID that we need to use. And then what we're gonna say is we wanna get the sequence in FASTA format. You can look up FASTA format if you want. It's just a specific format for RNA and DNA sequences that's easy to work with and is commonly used. And we're going to say we want the results in text format. And then what we'll do is we'll just return handle.read, which will give us back the sequence. Okay, so at a really high level, this function will take in an ID number for a nucleotide sequence and give us back the sequence. All right, so we're going to say sequence data is a dictionary, and then we're going to say for sequence in sequences, we're actually going to now download the sequence data. And we're going to say FASTA equals download sequence. All right, so what this is doing is it's going to loop through all of our sequences, and for each one, it's going to download the sequence from the NIH database and store it into this dictionary. All right, so this might take a little bit to run because it has to talk to an external site and do some downloading. All right, I will print this, but it's gonna be really long, so I will delete the cell after I do it. But you can now see we have the ID number and we have the actual sequence for the ID number, which is amazingly cool, right? To me, you can see the actual RNA sequence for the COVID virus, the first one that was sequenced, which is this. We're looking at it right now. And we also got the sequences for Delta and Omicron as well, so we can analyze them in just a second. All right, the next thing we're gonna do is, you may have noticed that the data is in FASTA format. We actually wanna parse that and just turn it into something we can more easily work with. So we're gonna use the FASTA parser from biopython. And that's in this seek.io module. So what we're gonna do is we're gonna loop through the sequence data dictionary. So that's what this does. So .items for KV and sequence data.items loops through all of the keys in the dictionary. The keys are the IDs, and the values, which are the actual sequences in FASTA format. And then we will basically create a file object using the FASTA sequence, and then we're going to parse that using the biopython parser. So we'll create a new key in our dictionary called parsed, and this will be the sequence that is parsed. So this is basically saying parse, we have to turn it into a file because biopython only works with files. So this is basically creating a fake file object with our string using this io.string method. Usually files are on a disk, but you can also create fake files in memory to kind of fool methods that expect a file. And then this is basically parsing the sequence out. And what we're going to do is actually, this parsing will create several different parse objects, and we only want the first one. So we'll just do that. io string io. There you go. Okay, so now in our sequence data, we now have a parsed field. So this is the FASTA field. Sorry, dictionaries are really hard to read visually when they have very long things. And then parsed is the actual sequence record from biopython that we can work with. So an easier way to look at this would be, let's go find this. Parsed. All right, so this is the actual parsed sequence record. All right, and what this lets us do is do some fancy analysis with these genomic sequences. All right, so the first thing we can do is try to align our sequences together. So let me go back to our presentation and just talk a little bit about what that means. When we have an original sequence, so let's say this is our reference sequence, original, and we have a variant, like for example, the Delta or Omicron variants, there's three types of ways that the sequences can change. There can be a deletion, so one nucleotide from the original sequence could be removed in the new sequence. There could be a substitution, so a nucleotide from the original sequence could shift in the new sequence, or there could be an insertion. So a new nucleotide could be added to the variant sequence. So that's how RNA strands can mutate, how the base of the base COVID virus can turn into Delta and Omicron over time through these different patterns of mutation. But what that means is we need to figure out where the sequences align and where they don't. So when there are mutations, you need to figure out which part of these sequences aligns to the other part and which part doesn't align. It's actually a really hard problem. It's tricky to actually figure out how sequences align. But our goal is to figure out which portions of the sequence align. Because if we can figure that out, we can figure out how different the sequences are. So that's going to be our goal. We need to figure out a score for how different the sequences are. So let's go back to our notebook, and I'll show you how to do that. All right. So we're gonna use an alignment algorithm, and we're gonna import that from biopython. And we're gonna create this pairwise aligner class that will basically help us align multiple sequences. And our aligner is using an algorithm under the hood to actually align the sequences, and that algorithm is called Needleman-Wunsch. It's a pretty cool algorithm. You can look it up if you want. This algorithm is basically how the computer can look at two sequences, RNA sequences, and figure out which parts overlap. All right, so what we can do is we can use the aligner to find a score. So what we'll do is we'll find the alignment between the reference sequence and one other sequence. So the reference sequence is going to be this. And we will then call the .seq property, SEQ, which gives us the actual sequence from the parsed data structure. So this is the parsed data structure, and we want to actually just get this sequence piece. We don't want the rest of it. And then what we'll do is we will look at the alignment with this sequence. Okay. All right, so let's run this. This takes a little while to run because how the algorithm works under the hood is building a giant matrix and then iterating through it, which takes a lot of time. But we can see our alignment score. So I'll talk a little bit more about how to interpret this score in a second, but this essentially tells you how aligned the sequences are in relation to the number of nucleotides in the sequence. So for example, we can look at our reference sequence. Our reference sequence is this long. So we can look at the alignment score based on the sequence, and we can see that these two are pretty fairly aligned, 99% aligned. So it's pretty close alignment between this, which is the reference sequence, and the first COVID sequence in North America. So the virus didn't mutate a lot between first being sequenced in China and then later being sequenced a couple months later in North America, which is really cool. We just looked at how a virus mutated over time, which it's so cool to me that you can access this data and work with it. It's pretty incredible. All right. Now what we can do is we can look at these scores between all of the sequences. So we'll create a comparisons matrix. I'll show you what this matrix looks like. So it's just a 4x4 matrix. And each matrix cell is basically going to be the alignment score between one sequence and another sequence. So we'll put all the sequences, we'll basically compare the sequences against each other and fill the values into this matrix. All right. So we'll loop from 0 to 4 because we have 4 sequences. So this will essentially compare every sequence against every other sequence. And then what we'll do is we will do the same thing we did before, except instead of this, we will do sequences I, and then sequences J. All right, so this will basically loop through our sequences list twice, and then compare each sequence with each other sequence. That's what this loop is doing. And then we will put the result into our little comparisons matrix. All right, and then we can turn that into a data frame. This is going to take a little while because it has to compare all of those sequences. So we're essentially just going to create a data frame. We created a NumPy matrix, but a data frame is just a little bit nicer to work with. All right, and then we will assign human names as the column names and the index names, so the column and row names. And then let's take a look at this. Once it finishes running, this could be a little bit, we will go ahead and take a look at how all of these compare to each other. All right, so we can see the reference compared to the reference is 100% match. These other numbers are a little bit harder to interpret, but what we'll go ahead and do is just go ahead and divide the first row of the comparison matrix. So that's what this ILOC is doing. This is grabbing, sorry, the first column. This is grabbing the first column of the matrix. And we'll just divide it by the length of the reference sequence. So this will just give us the kind of percentage overlap between each of the variants and the reference sequence. So we can see the base was the sequence from North America 2 months after reference sequence. So the overlap here is very high. The delta sequence overlap is still very high, 99.7, but a little bit lower. And then Omicron has diverged a little bit more. So as time goes on, it seems like the virus is mutating more and more, but it's not mutating all that much, right? I mean, I don't know how much viruses typically mutate, but the sequences are very similar. All right, okay. So now we can see basically the percentage overlap. Our next step is actually going to be look at the mutation points. So let's grab a couple of sequences, and then we will look at these sequences in a little more depth and look at the actual mutation points. So let's look at our reference sequence, and let's also look at, let's look at delta. So that will be this really long string. Okay, so we're going to grab these two specific sequences, and we'll just grab the actual parsed sequence. All right, and then what we can do is we can use another biopython method called align to actually find the points of mutation, insertion, deletion between these two. And then let's take a look at delta alignments. So we can see we have this pairwise alignment object. This object is actually a list. So we actually just need to grab the first one. So we'll assign that to delta alignment. And what we can do then is just look at delta alignment.shape. So this shows us, okay, we aligned two sequences and there are 29,943 nucleotides that we aligned. And then we can take a look at the points where the sequences are aligned or not aligned. This is a little bit hard to interpret, so I do have a slide for this, but I'll show that in a sec. But basically what this is this is a tuple of tuples, or we'll just think of it as a list of lists, it's easier. The first list is this, and then the second list is this. And basically what it does is it shows you the overlaps between the sequences. So the first item in this first list is this, and this first item in the second list is this. Those are the parts of the sequences that align. Let me show you a slide to make that a little bit more clear. Okay, so basically there's two lists. The first list basically lists out all of the points of alignment in the first sequence. The second list lists out all the points of alignment in the second sequence. So you can see 0 to 209 align between both sequences. 210 to 212 aligns with 209 to 211 in the second sequence, etc. There are some missing values. There aren't in this example, but missing values are points of deletion on one side, insertion on the other side. So we can actually iterate through this list to actually figure out where the insertions and deletions are. All right, let's go back to the notebook and let's do that. All right, so the first thing we'll do is we will actually loop through. We'll loop through these and then we will print out all of the mismatches. All right, so the way we'll do this is we'll say for alignments in zip. Zip is a very cool function if you haven't used it. it basically, so we'll do delta alignment, dot aligned 0. All right, so this is the alignments from the first sequence. This is the alignments from the second sequence. Zip basically takes the first element from both and lumps them together into one list, takes the second element from both, lumps them together, and so on. So alignments here will be the first time it'll be this tuple and this tuple. The second time it'll be this tuple and this tuple, and so on. So it gives us the alignments between both sequences. We're also going to create variables called sequence1end and sequence2end. And we're basically going to loop across all of the alignments and print out any points of mismatch. So what we'll say is if seq1end and seq2end, the end is going to be the end of the last alignment. So what we'll say is the mismatch all right, this is a little bit complex and I do not have a diagram for it. In retrospect, I probably should have created one. Basically what this is doing is this gives us all of the alignments. What we care about are the misalignments. So this will basically give us the inverse of the alignments. It'll give us all of the misalignments. So we can print out, hey, where do these two sequences not align? All right. Okay, and then what we do is we just print out the mismatches. So we can print out the mismatches in 1. And then we can print out the mismatch in 2. All right, and then what we do is we just assign seq1end to alignments. This is the end of the previous alignment. Okay, so basically what this is doing is this is looping through all of the alignments. It's saying if it's the first alignment, skip this, and then figure out where the end of the alignment is and assign it to this. And then anything from the end of the previous alignment to the start of the next alignment, we're going to print out because that's a mismatch. There you go. I missed the N. Typos. All right, so basically this shows you all of the mismatches. And there's not a lot, but it doesn't show you where the mismatches are happening. So the next thing we can do is just smarten this up a little bit to actually show you where the mismatches happen and color code the type of mismatch. All right, so the next thing we'll do is actually show you where the misalignments are happening. And a thing we can do to color code them and make them easier to see is use the IPython or Jupyter. IPython was the previous name, or IPython and Jupyter have overlapped in a lot of ways and haven't overlapped in ways, but IPython is now a core component of Jupyter. But it can basically let you display data in HTML format. So that'll let us color code the mismatches. So we'll create a function called colorPrint, which will basically just display data in color for us. And our default color will be black. And what we'll do is we'll just style this with some HTML. I know inline CSS, for anyone who's familiar with HTML, is the worst, but we're going to use it anyways, because we have to. All right. So basically what this will do is it will return HTML, and it will display that HTML in Jupyter Notebook in a specific color, which will let us see where any mismatches or anything else are happening. So I'm just going to copy this, and we're going to improve this, this existing sequence of code. And what we'll do is show the type of mismatch. So basically, in order to do that, what we'll say is we'll check the lengths of the mismatches. So if sequence 1, the mismatch is 0, and sequence 2, the mismatch is several characters, then it's an insertion. Other way around is a deletion. If they match in length, then it's a substitution. So what we'll say is, actually, let me do this further below. We'll say if the length of the sequence 2 mismatch is 0, then we know this is a deletion. So what we'll do is we'll do color print seq1 and yes, we'll just do color print seq1 mismatch, and we'll print that in red. And one thing is this color print won't actually print it. We have to separately display it. So I'll just create a list to keep track of all of the things we need to display, and we'll just append this to the list. All right. And then if the length of the sequence 2 mismatch, or sorry, sequence 1 mismatch, then we know it's an insertion. So what we'll do here is just add in the sequence 2 mismatch in green. That's an insertion. And then otherwise, it's a substitution. So what we'll do is we will add in some code to deal with the substitution. And what we'll do is we'll just add in the sequence 2 mismatch, but in blue, because that's a substitution. Yes, this should work. Okay, and then in cases where there is no mismatch, we still need to add in the regular alignment. So we'll just add in sequence 1. So we'll just add in the length of the alignment as well. So this will just give us the full sequence. So that'll run. And then the next thing we need to do is we need to join our display sequence. First, we need to convert them all to strings. So let's go ahead and run that. And then the next thing we'll do is we'll go ahead and display our HTML. So we'll display this in HTML format, and we will have line breaks between all of the different components. So we'll go ahead and join our display sequence. All right, and let's run that. Okay, so now we can see the full sequence. So these are in black is where the sequences align. The red is a deletion. So this is cases where something from the original sequence was deleted. In this case, the reference sequence was deleted by the Delta variant. Green is where something was inserted by the Delta variant. And then blue is a substitution. So you can scroll through and you can see exactly where those occurred. All right. But there are a few things you can do if you want to continue this analysis. So a couple of interesting things you can do are you can convert the nucleotide sequence to codons and then figure out which proteins those are encoding. And you can determine how that shifted between variants. You can also look at some other variants, and you can look at mutation over time because a lot of the metadata has timestamps. So you can look at how mutation patterns and how insertions and deletions shifted over time. You can look at the most common insertion, deletion, and substitution sites to see specifically which ones are changing the most rapidly or least rapidly. So those are a few things you can do to follow up on this. But there's a lot of fun analysis you can do, and it's still really cool to me that you can even access this data in the first place. Yeah, we just looked at the actual genome of the COVID virus. If you want to go really advanced here, if you look in that COVID RNA folder I shared, let me actually find that. There is an implementation of the Needleman-Wunsch algorithm for alignment. It's kind of advanced, but it's always fun to try to implement these kinds of algorithms. So feel free to take a look at that if you want to try to implement it yourself. Okay, thank you so much, everyone. I really appreciate it. Have a great rest of your day.